Hey guys, today we're going to talk a little bit about viruses. So, a virus is a non-living particle. It is made up of nucleic acid, so DNA, and either a protein or a lipid protein coat called a capsid. So here we have your doodle notes already done for you, okay? So, viruses are very, very, very small. They're even smaller than prokaryotes. They don't have any cytoplasm or organelles, so they don't carry out normal functions like metabolism. Now, viruses cannot grow or divide on their own. They have to have a host cell. So, to reproduce, they enter the living host and use their ribosomes, enzymes, ATP, and other molecules to reproduce. Because of this, they are not alive. There are a bunch of different viruses that you have probably heard about, such as the flu, the common cold, Ebola, and now, of course, COVID. So, viruses can be bad, but they're also useful tools for genetic research. So they can be used as vectors to carry good genes into a human cell because they have to have them as a host. So to do this, a scientist would remove the genes in the virus that cause diseases. They would replace those with the genes that they want going into the cell and insert it into a cell and allow the virus to insert its genome into the host and then make the desired changes. So let's see, back to this. We have a protein coat or a capsid that surrounds the virus. Some viruses infect bacteria, and those are called bacteriophages. Now, when a virus enters a cell, reproduction can happen in one of two ways. If the virus copies itself immediately, it is called a lytic cycle. If it is inactive for a while in its host before it replicates, that is called a lysogenic cycle. So what is the difference? In a lytic infection, a virus will enter a cell, make copies of itself, and then cause the cell to burst. So for example, a bacteriophage, or a virus that infects bacteria, will inject its DNA into the bacterial cell. The bacterial cell will make messenger RNA, which will turn into proteins, and it will chop up that cell's DNA. Now the host makes new virus particles, and before long, it's full of these particles and the cell will burst open. Now, in a lysogenic cycle, the host cell is not immediately taken over. Instead, the virus is going to insert its nucleic acid into the host cell's DNA. That viral DNA will be copied along with the cell's DNA. And the cell is not damaged at this point. However, daughter cells, so once they divide, will be infected by this virus. So, inside living cells, viruses are going to use their genetic information to make many copies of themselves. Some replicate right away. Other viruses stay inactive for a long time within a host cell. So, one virus that you might get even every year is called a common cold. So here is what happens. A capsid, or the virus, will land on a cell, often inside of your nose, and it's taken into a host cell. Inside that host cell, a viral protein will make a whole bunch of new copies of its RNA. The ribosome in the host will mistake the virus's RNA for its own. So the viral RNA, or the viral genetic information, is translated into capsids and other proteins. New virus particles form, and within 8 hours, that host cell bursts. When the host cell bursts open, hundreds of new virus particles can then infect other cells. So these viruses, to grow and reproduce, have to infect living cells. They rely on the nutrients and the other machinery of these host cells, kind of like parasites. Matter of fact, viruses are parasites. Now, they're not alive. Even though they have characteristics of the living things, they cannot reproduce on their own, so they are not considered to be alive. Now, that is all. Thanks, guys.